will get going with the introduction here. Let me just, there we go. Great. For those who don't know me, my name is Eric Sperling at Stanford, and I'm really excited today to be presenting this proxy primer on iodine calcium through the Sedimentary Geochemistry and Paleoenvironments Project, or SGP. If you're not familiar with SGP, it's an open community-oriented research consortium that seeks to address questions of environmental evolution across Earth history, primarily through statistical analyses of the sedimentary geochemical record. And so we are not just a database project, we are a database project that then tries as a group to interrogate this, this large sedimentary geochemical record. We have more than 140 people around the world that are involved, and we hope if you're not involved and you're on this call, you will be involved too. So we developed the Proxy Primer series as a series that can accessibly explain the basics of a geochemical proxy or method to a broad audience, particularly because we do have a relatively broad audience in SGP. And the idea was to curate these lecture videos on our YouTube channel to create a proxy video library that people can go back to as they have new students that are coming through or just want to learn about a new proxy. And also to provide an open space for interaction with the experts on each proxy. So just looking at our 2022 schedule, today we're hearing from Dr. Zunli Lu on iodine calcium. We then have a big break for the summer, and we are coming back in the fall with a fairly packed schedule with Dr. Maya Gomes talking on sulfur isotopes, an in-person meeting at GSA, and then finally Dr. Selva Marroquin talking about carbon isotope chemostratigraphy to close out the year in November. So I'm delighted today to introduce Dr. Zunli Lu. Zunli did his undergraduate degree in Nanjing before moving to the University of Rochester for his master's and PhD, and finally on to Oxford for his postdoctoral studies. He then started his faculty position at Syracuse, which is where he's been ever since. And in particular, he's been involved in developing the iodine calcium redox proxy, which for me as a paleontologist, I've always been really excited about because I think a lot of the redox proxies that we use, so for instance, I've done a lot of work on iron speciation, it can be good at telling you between oxic or anoxic bottom waters or ferruginous or euxinic bottom waters. But it really has a hard time telling you about the spread of low oxygen conditions. And for animals, animals can live in well oxygenated waters. They simply can't live in anoxic waters. And most of the interesting biology actually occurs in between in kind of the spread of low oxygen or dysoxic waters. And so I think particularly from that perspective, although just kind of more broadly as a really solid carbonate geochemical proxy, iodine calcium is really exciting. And so I'm excited to hear more about it today. So with that, I'm going to stop sharing and have Zunli tell us about the proxy.
Okay, so the screen seems to be working.
Yep.
Okay, let me turn on the pointer. All right, Eric, thanks for inviting. And as I was preparing for this talk, I've been thinking about the sort of the history of the proxy, and since we started working on it, it's been more than 10 years. But actually it's not that well-known proxy, and particularly for this kind of talk, I think I'm gonna sort of do three parts. The first is for more tuned towards someone who has never heard of this proxy and never thought about iodine. Then I'll get into a few selected proxy applications for those of you who have been tracking this proxy or have read a few of our papers in detail and maybe have more deeper thinking about it. I have a third part, so we'll get into a little bit more details.
Thanks.
Um,
I'm also like in a zoo. It's fine.
Sorry, Amy, could you please mute yourself?
But it's okay. I'm just listening.
I can mute her. Sorry about that.
no problem. Yeah, it happens.
Yep. All right. Good to go.
Okay. So first of all, what's the basic idea behind this proxy? It really boils down to the redox geochemistry of iodine. So if you look at this graph adapted from an older paper, you have a log concentration on the y-axis and then the PE on the x-axis. So the general idea is if you have oxygenated seawater, the majority of the iodine, most likely you'll see iodate, IO3 in the water. And then if you have anoxic seawater, most likely you'll run into iodide, which is reduced species. These whole bunch of other interesting redox chemistry that can happen. You don't, for the purpose of our proxy, the concentration is too low. These things in the seawater, so we kind of don't have to worry about them. And when I was doing a postdoc with Ross Rickaby at Oxford, we were working on some totally different things. And then this iodine proxy was actually a side project. At that time, because I did my PhD on iodine isotope, and I was surrounded by people working on these paleoproxies, so I started thinking, well, maybe we can actually use it as a proxy. And so the first thing I decided to do is to see if it can get incorporated into carbonate. So what you're looking at here is we made some calcium carbonate in laboratory condition, and we spiked the solution, the medium, with iodide in one series of experiment, and spike the solution with iodate in another series of experiment, and then we measure the iodine calcium ratio in the calcite. So the result is we see that iodate gets incorporated into the carbonate, but iodide doesn't get in there. The very, very basic idea is if we get some carbonate that grows in the seawater that records the iodate concentration, then we can use this to derive some information about the O2 level in the water. So next, I want to show you how the iodate profile looks like in modern ocean. This is a location where you have the oxygen minimum zone. So O2 goes down very rapidly into the OMZ. Similarly, you see the iodate also crashes when oxygen crosses a threshold. And then it will gradually recover into the deeper part of the ocean. So if you look at a typical iodate profile like this, you have sort of three parts in the surface water. Iodine can be taken up by organism as a micronutrient, and then in the middle part, iodate can be reduced, just a redox reaction. And then in the deeper ocean, because iodine has relatively long residence time, you will see in most part of the deep ocean today, the iodine or iodate in this case, concentration about 0.5 micromole. For those of you studying carbonate rocks, especially in relative deeper part of the, especially in the deep time, we're mostly looking at, or we're mostly sampling this upper ocean signal. In a map view, this is an example of a modern ocean hypoxia. Here you can see where you have a very intense biological productivity, you end up losing oxygen, creating shallow, low oxygen water in this corner. And then what I actually want to point to is these values are all surface water iodate. So you can immediately realize that if we have carbonates growing in this area, you will get very different iodine calcium values. And this immediately shows you that we're looking at a local proxy. And second thing I want to sort of point out is that all of these iodine signals that we're looking at in the ocean, in this case, can happen at the same atmospheric oxygen level. So it, again, it has some implications when we deal with really deep time. Okay, so that's in the second part. We're gonna look at a couple of, well, a few case studies, how this proxy has been applied. When I guess my thought process of selecting these case studies is that I want to cover different age from very deep time to relatively recent or closer to the modern ocean. And then I want to cover different durations, meaning some of the events may occur at 1,000-year time scale, and then other studies that we're going to look at may cover a really long time interval. Also, I want to show you how this proxy does in terms of different oxygen changes. So you can see, you can use this proxy to capture O2 rise, oxygen rise in a really low atmospheric O2 world. You can also use this proxy to capture the deoxygenation events in the ocean in a high PO2 world. You can look at long-term evolution. You can also play with the paleoceanographic conditions with this proxy. So I'm going to try to show examples that cover all of these different types. If we want to really go dive into the deep time and see whether this redox proxy does what it's supposed to do, it doesn't get better than the GOE. So as soon as I published first paper about this proxy, Tim Lyons' group got interested, and we started working together. And we first look at these really old rocks where the whole world's supposed to be anoxic, or mostly. And then the big change happened during the great oxidation event, where atmospheric oxygen started to appear and accumulate. And indeed, we see the first iodine signal, detectable iodine signal in the carbonate rock right during the GOE, and we couldn't get any iodine signal in the rock before that. So at least to me personally, what's I guess a little special about this record is iodine is a direct evidence of dissolved oxygen in the seawater. So the next case study I want to show is this Mesoprotezoic, the Boring Billion period. After the GOE and the Mahmud-Gandhi high iodine peak, we get into this relatively low value of iodine numbers throughout for more than 1 billion years. The only exception is that at that time, Dalton was not very excited about seeing these relatively high values in middle of the mid-Proterozoic part. And it looks like it's not the only case. Another group independently found some higher values at a slightly older time interval. And then this is the mid-Proterozoic part is not my personal strong interest, but I sort of find it a little bit intriguing that iodine signal seems to point to a little bit of a oxygen action in the during the boring billion. But there are also other groups using different approaches that seems to draw the similar conclusions. As we move on through the Earth history, the Neoprotozoic is a really fascinating time interval. So what I got interested in, one of the things is the bitter spring stage, kind of because a lot of important animal groups sort of trace back to the origin in this time interval, roughly around here. So what happened to the iodine? Working with the Galen Halverson's group, we put together this this iodine record. There are a lot of carbon isotope fluctuations here, and this is the main bitter spring stage. And we found a really big iodine spike. This was the first time we saw these values in the Precambrian rocks, and we got very excited. And then not only the iodine spikes up really high during the sort of the recovering or the rising limb of this C13 excursion. If you look at throughout this record, you have multiple like these C13 values that drifts up, and then most of the Precambrian iodine values, iodine calcium ratios, are below half micromole per mole, and all of these sort of sporadically high iodine numbers were more frequently observed during the positive drift of the C13. So that's something we also find quite fascinating at that time. And then we published this paper, and it was quite interesting to see the iodine spike was reasonably close to the chromium signal at around this part of the event. So my strong interest in the Earth history was mostly in the Phanerozoic. And when I was a grad student, the geocarb was pretty much shaped how I think about the global ocean and the atmosphere at that time. And then, well, according to the model, the atmosphere has been pretty much very well oxygenated, just like modern days. And then, so the question was also what happened in the ocean, and that's what I was very curious about to try to find out, especially using the iodine proxy. And these are the couple of examples of papers that got me thinking or got me questioning a little bit of what the geocarb model may be representing. And then there are both proxy data and also other considerations about the model that think there may be some quite a bit of uncertainty in our understanding of the especially the Paleozoic ocean. So it took us quite a few years to collect a larger data set of the bulk rock iodine calcium all the way through Proterozoic, Paleozoic into the Mesozoic and Cenozoic. What you're looking at here, these boxes shows the majority of the values from that one section. And then you have the minimum and maximum values at these individual sections. So immediately the observation is these boxes, or the majority of these values from a section sits quite low during the Proterozoic. But as we get into the Paleozoic, we start to see these boxes that sits a little bit higher. And as we move on to the Mesozoic and Cenozoic, majority of the background iodine calcium values were quite high. If you look at the distribution, interestingly, the Paleozoic iodine calcium distribution is more similar to the Proterozoic, and Mesozoic and Cenozoic has a very different distribution. So how do we interpret these long-term shifts, right? So that was what I was very interested in. That end up in this paper, we combined the data interpretation with the Gini Earth system model. So what we're looking at here, there is sort of a high spike in the iodine values in the Paleozoic, more specifically during the Devonian, and then as we move on to the Mesozoic, we see this background iodine calcium value took a step change and getting to more like the modern condition. How do we interpret that is the iodine calcium, especially these bulk rock values, they more or less reflect the upper ocean condition, which is kind of the sandwich between the atmosphere and the intermediate to lower part of the upper ocean. So if there's a OMZ here, especially if there's a shallow OMZ, even if the PO2, even atmosphere is very well oxygenated, you can still get very low iodine calcium signal just in the modern day OMZ. And then if we look at vast majority of the ocean today, we have more or less like this condition, well oxygenated and with the iodate values that are relatively high throughout the water column. Occasionally you get some OMZ popping up around the continental margin. So in terms of the temporal trend, so we think there is a Paleozoic PO2 rise. So the atmospheric PO2 likely spiked up during or took a step change during this time interval, which possibly drove the iodine calcium to go up. But if the OMZ, if there's a shallow OMZ ubiquitously occurring around the continental margin, then our proxy will still capture these relatively lower values. Now, until the shallow OMZ start to disappear or become less frequent, then our iodine calcium can sort of show these relatively higher background values. And what controls, what may control the OMZ is the remineralization of organic matter. So surface water productivity will give you a lot of these organic matter. If these organic matter doesn't sink through the water column very fast, it gets decomposed within the relatively shallow part of the ocean, then you're going to end up with these shallow so-called oxycline in a lot of places around the coastal regions or around the continental margin. But if we have these modern marine organisms start to appear, like forams and diatoms, and these things, they biomineralize, and then they sort of go through ballast. There's stronger ballasting effect, so they pull their shell will pull these the organic matter through the water column much faster than in the older days, especially during the Paleozoic time. And this potentially can get rid of these ubiquitously shallow ONZ. So that's sort of our general interpretation of this this long record. And the next, I'm going to try to show you some, as we gradually move into a high PO2 world, the ocean can still lose oxygen periodically, and that will result in some of the possibly mass extinction events. So these is an example of the early Ordovician mass extinction. So what we're looking at is during a carbon isotope excursion, we see the iodine calcium crashes down, and then possibly you have additional other redox proxies supporting that we have a deoxygenation event occurring here. Very similarly, this is another major mass extinction occurred at the end of Devonian, roughly around this time. So very similar temporal trends that we're seeing. There's a major carbon isotope excursion, and then you see the iodine sort of crashes down to very low values. In this particular case, about the Hagenberg, what I liked about this iodine record is iodine stayed fairly low for a relatively extended periods of time. And if you look at the fossil record, the Hagenberg is a fairly stretched crisis in terms of the time interval. And there were some indications early on used to link this event to anoxia was the black shale, but the black shale only occurred in short interval. So this potentially can show you that the oxygen level in the water actually remained fairly low for relatively long periods of time. And then this is a paper that just published about the end of Triassic. Again, similar story. You have a background value of iodine calcium. It's relatively high, and then during the extinction, you see the iodine crashes down. Again, in this case, these folks try to compare the iodine as a local record to sulfur, which has more indication of the global conditions. From there, I want to show you a little bit more how we can potentially use iodine to look at a spatial pattern in the ocean. This is a study that we did for the Mesozoic oceanic anoxic event for the Cenozoic and for the Cenomanian Turonian transition. We looked at, we first got a few stratigraphic trend from multiple sections, just like what I showed you in previous studies. And one thing that we noticed is the background values or the iodine calcium values before, during, and after the OAE are showing these three bars sort of stacked together. And then you can see there's a pattern, there's a spatial pattern. In some parts of the ocean, the iodine calcium are generally high, but in other parts of the world, you get generally low iodine calcium throughout the event. And we have some O2, the upper ocean oxygen levels that were simulated in Gini, and you see there is sort of a match between the simulated O2 versus the measured iodine calcium. So this is actually the first study in relatively deep time that we saw there's a potential that we can compare the iodine calcium spatial pattern with Earth system models. So in this paper that we recently published, this is about the late order vision mass extinction events. In this event, we have two extinction pulses, so-called LOME 1 and LOME 2. What got me excited about this record is we were generating iodine calcium records from the same samples that were used to generate this uranium isotope record. I'm sure at some point you'll hear a uranium talk in this series. But the idea, general idea is here the uranium is pointing to a expansion of anoxia on the seafloor during the LOME 2. And this is the second extinction pulse. But right in the same sample, we actually see iodine calcium sort of popping up. So that the immediate interpretation is we have local upper water O2 that were actually going up during this extinction, whereas the global proxy seems to indicate an expansion of anoxia on the seafloor. So this is a very interesting to try to reconcile. And at the same time, independently, Alex Paul, working with Andy Ridgewell, has been modeling this event. And what he discovered is in order to simulate the increase or the expansion of seafloor anoxia, there are actually different ways that you can reproduce that anoxia in the model. So you can have PCO2, this on the x-axis that shows the PCO2. If you cool the ocean from a relatively high PCO2 level without changing the phosphate, the nutrient level, you can, the model says you can increase the anoxia from almost nothing to 50%, roughly half the seafloor, to become anoxic. And another way you can produce this kind of signal, as indicated in the uranium, is you can cool the ocean at the same time and bump up the phosphate. If you increase nutrient level, you can get similar degrees of anoxia expansion in the ocean. But what happened in the upper ocean is where iodine calcium becomes helpful. So these two scenarios, they can give you similar degrees of anoxia expansion in the deep ocean. Actually will give you opposite O2 changes in the upper ocean, which is shown in here. So in one scenario, your O2 actually goes up. So the way you want to read this graph is if it's warm color, here it shows the O2 in the upper ocean increase during this event. And then this is the opposite. In another scenario, your upper ocean O2 actually decreased during the cooling. This is mostly driven by the nutrient. But I just want to show these couple of studies to show you that you can really start using iodine calcium and sort of simulate a little bit of a paleoceanographic changes. You can read the details in the paper. As we move on into more recent time scale, when we started develop this proxy, I really wanted to apply this in glacial interglacial time scale. So this is a fairly recent time interval. So we had a core from very high latitude Southern Ocean. Today, this area is very, very well oxygenated. During the glacial, interglacial cycles, at this location, the planktonic foranes shows that iodine calcium value actually dropped below certain threshold. That shows that we potentially have O2 that is noticeably lower than today. A more interesting thing to me at that time was if you look at the iodine calcium, the time when iodine calcium drops below the oxygen threshold. And this is also at the time when the PCO2 in the atmosphere dropped below this long-term average. So Southern Ocean definitely plays a bigger role, important role in adjusting the CO2 level in the atmosphere. And then later, there were also other groups using different proxies. They were also saying this AAIW potentially gets fairly, the O2 gets fairly low during the glacial. So this is another study that we did for the glacial and the glacial time. With the availability of forams, and we can really start getting these records for different parts of the oxygen profile. So in the upper part of the OMZ, we got the iodine calcium values throughout these intervals at different locations. You can get to the core of the OMZ. You can go slightly outside. Vertically, we can also have additional foram proxies that can be quantitatively converted to bottom water oxygen throughout this time interval. So with the foram, and if we know where these forams live, we can actually sort of constrain this O2 profile vertically. This vertical profile, we can get a better idea of the shape of this profile through time. One of my PhD student sort of combine iodine calcium with the more foram proxy. In this case, what you're looking at is sort of the modern calibration. You have oxygen, the bottom water oxygen plotted on the x-axis, and then you have this C13 gradient proxy. Basically, you have two forams. One live at the sediment water interface, or slightly below that, and then you can have another species that lives at the oxic anoxic boundary in the pore water. And if you subtract the C13 delta C13 between these two foram species, you can calculate, or you find a correlation between this kept out C13 with the bottom water oxygen. So here you can really be a little bit more quantitative about the bottom water O2 value. And then on the other hand, if you look at these forams, when the O2 gets really low, you start to see these foram to grow a lot of holes on the surface. And then if you can process these image and come up with these pore density, the density of these holes on the surface of foram, you also can be saying something a little bit more specific about the O2 values. So iodine calcium, especially benthic iodine calcium, can be compared with these two independent proxies to give you a little bit more quantitative estimate of the glacial ocean. So you can read the details in here, but for this group, I just want to briefly mention that iodine calcium, especially benthic iodine calcium, is in a really good company in this time interval. So I don't know how we're doing in terms of time. Now we're gonna, I want to show a few slides about some of the details, some more, I guess, considerations if you really want to use the proxy, and there are a few things that you want to be aware of and be careful about. The first thing, especially in deep time, the first thing we want to worry about is diagenesis. So how does iodine calcium do in terms of diagenesis? This is one example of a meteoric diagenesis. What you're looking at is we're going across certain transition. Here, some of these corals have been meteorically altered, and you can see iodine calcium either it goes down or doesn't change. In this case, say at the same time you see the C13 gets altered. Similarly, if you want to look at marine burial, iodine calcium also go down during the diagenetic alteration. So far in the Dalton Hardesty did, if you want to look at more of this kind of work, go to this paper, and Dalton looked at a whole bunch of diagenetic scenarios, and all we see is iodine loss from during the diagenesis. So far, we've not seen any case where secondary processes would increase iodine calcium. So that's what we know so far. And for the deep time studies, it's impossible to eliminate the possibility of diagenesis. So you really want to be careful how you interpret the iodine signal, in particular the absolute value of iodine calcium ratio. And as a sort of very basic consideration, we look at thermodynamics and say, okay, iodate is dominant in the oxic seawater, and iodide is dominant in the anoxic water. So if we, the reality is always a little bit more complicated. So if you look at a whole bunch of data that's collected by chemical oceanographers, what you're seeing is generally you have the pattern that you have a lot of scatter in the high O2 world to intermediate O2 range. And then at some point, once oxygen gets low enough, iodate will start crash. You have very low to near zero iodate. But you also have some of these low iodate values in relatively high O2 water. So these is mostly because of the kinetics of the iodine redox reaction. So you have iodate reduction, and you also have iodide oxidation. And both of these reactions are heavily mediated by biological processes. And so you may have a slow oxidation in the iodate, which will give you these conditions here. And then you get slow iodide oxidation. And then iodate reduction in most cases is relatively fast once it kicks off. The reduction of iodate is fairly fast. So what's the implication of the kinetics issue when we actually look at the rock records? So immediately you want to think how local, is it really local or is it regional signal? So this is some of the COTOP study that my student did. So if you look at these in the map view, and these planktonic forams, they generally show high iodine calcium in high oxygen waters. They show low iodine calcium in low oxygen waters. But if you actually plot them in this fashion, you'll actually notice again we have just like here we have low iodate in high O2 water. We actually did see some low iodine calcium in relatively high O2 water. So this is when you plot strictly the iodine calcium value from the O2, the minimum O2, the lowest O2 value right at this site. But if you plot the iodine calcium against the lowest O2 in a quarter degree grid, you immediately, these data points will shift towards the left. What does that mean is basically you have low oxygen water near these sites. They're not necessarily directly below where these forams were growing. So if you have these water mass advection, you can locally capture, and also the slow iodide oxidation, you can get some situations, get into situations like this. So if you want to, if you're a paleoceanographer, you're very used to look at one foram data and say this is exactly, try to pinpoint the water condition right around this foram habitat, that you need to be a little bit careful when you use this this proxy. So okay, the next thing I want to quickly mention is how to really interpret the low values, low iodine values. So this is the first sort of quartile data set that we did in 2016, and this is what we did in 2019. So it's still, you see a lot of scatter in the high O2 range. And then as soon as you cross a certain O2 threshold, at least in the planktonic foram, you see also a lot of scatter. So to me, this is best used as a semi-quantitative proxy in foram at least. If you look at some of the recent data that my student generated when he is now at Woods Hole, this paper is coming out, and this is the benthic foram iodine calcium, the foram that lives at the seafloor. Again, you see a lot of scatter at the higher bottom water oxygen range. You see a lot of scatter, massive scatter. As you go into lower O2 range, iodine calcium generally stays fairly low. There's always the temptation to put a line to fit a equation through this this kind of a quartile calibration. You can try to do that, but I think it's really problematic if you try to apply these equations back in the rock record or in your downcore record, because there are a lot of scatter, and there's also because of this threshold behavior. So at least for me personally, I don't see an easier way to use iodine calcium as a fully quantitative proxy, at least not across the entire O2 range. Later on, when this paper comes out, you can see that there are additional things that we need to worry about when the O2 is relatively high, and iodine calcium here, the scatter here is not driven by oxygen. So if I want to give a quick summary how I would use this proxy, so first of all, if you look at bulk carbonate is what you deal with, the stratigraphic trend is something I would focus on, especially if you have a drop in the iodine calcium in your stratigraphic trend. That's something is relatively useful if you can do careful diagenetic screening. And when you're dealing with forams, you can use those iodine calcium thresholds that are being proposed here. I think these thresholds, if you use them in 4M, you can say something about, you can put some values of the dissolved oxygen in the seawater. You can sort of link these low iodine calcium value with certain dissolved oxygen value in the ocean. But I would most likely use this in the 4M in more recent time scale. I don't want to stretch this or use this threshold value in the in bulk carbonate rocks from deep time. I think that is potentially problematic. And if you want to say something about the O2 value, I think most likely this proxy will be limited as a semi-quantitative proxy. What are the things that I'm starting to see that I think probably folks should be really careful about doing? The first thing is try to fit some kind of regression line in these quartile data and across the entire O2 range. So if you try to fit a line, you'll get R square that may not look too bad. But actually, if you go back and try to apply this kind of a equation in your downcore record, you will very likely get the wrong answer. And in terms of relatively deeper time, if you try to use the O2 threshold that we came up in the 4M, if you try to use that in bulk rock like these, you really want to be careful. And personally, at least in the paper in my group, we haven't tried to say anything about the dissolved O2 value in these deep time bulk rock. The last thing that I think folks should be extremely careful about is to use iodine calcium to derive some atmospheric oxygen level. So when I see a sentence like this in a paper published saying you can sort of infer atmospheric oxygen level from iodine calcium, to me this is pretty alarming. The reason is fairly simple. If you look at the modern day iodate distribution in the seawater, you get huge variability from places to places. And this is not the most complete compilation, but it gives you the idea that when you go back in time, especially go back billions of years, you take one core and measure these iodine values and try to calculate the atmospheric PO2 value. That is something extremely difficult to do, in my opinion, nearly impossible. So these are the things that I thought if you're interested in using this proxy, you want to be careful about how you interpret the data. I guess you can, whether you look at the Elderfield curve of the proxy confidence, or you look at some sort of cognitive process of the human brain, or whether you want to look at sort of investing in this proxy and sort of you want to draw the analogy to the stock market. Where this proxy is currently, it's hard to say, and in my view, it really depends on who you're talking to. Individual researcher may be at different parts of these curves, different stages. So I guess I'll wrap up my talk here and take some questions if there are any.
Great. Thank you so much, Zunli. That was excellent. It was great seeing all the range of things that you've applied this to, from paleoceanography to deep time and so forth. I'm going to turn off the recording.